goldfish swims into a chamber, a green light comes on, and it crosses to the other side before the shock ever arrives. It has learned this perfectly every time. Then a surgeon removes a single, specific patch of tissue from its brain, no larger than the head of a pin, and returns the fish to the exact same test. The light comes on. The fish does not move. The shock arrives, and the fish takes it, exactly as if the danger had never been taught to it at all. That is not a simplified version of the finding. It is the finding. In 2004, the researchers Manuel Portavella, Blas Torres, and Cosme Salas published this result in the Journal of Neuroscience, in a paper called Avoidance Response in Goldfish, Emotional and Temporal Involvement of Medial and Lateral Telencephalic Pallium. The training was simple and consistent. A green light warned the fish for up to 15 seconds. If the fish crossed a barrier within the first 10 of those seconds, nothing happened. If it did not, a mild shock followed for up to 5 seconds. Goldfish learned this fast. Once they had, the surgery began. One group of fish lost a specific region called the medial pallium. A second group lost a different nearby region, the lateral pallium. Every fish then went back through the exact same test it had already mastered. The medial pallium group lost the avoidance response almost entirely, a result the researchers describe as considerable impairment in the retention of a response the fish had already learned, tied cleanly to that one region and confirmed statistically. The lateral pallium group told a different story, largely keeping the avoidance response while losing ground on a separate, more time-dependent version of the same task. Two neighboring regions in the same small brain, running two different separable jobs. Damage one, and the fish forgets to be afraid while remembering everything else about how to swim, feed, and navigate its tank. Damage the other, and something else breaks instead. A brain that built one general-purpose memory system for everything would not need two regions like this at all. The reason to split fear specifically into its own dedicated hardware, separate from ordinary memory, is that fear has a different job to do. An ordinary fact can sit quietly until it is useful. A fear has to be available instantly, without waiting for deliberate recall, the next time anything resembling that warning shows up. Building that as a separate, fast-access system, distinct from the slower system that tracks plain facts, is a specific engineering choice, and it shows up as two separable regions doing two separable things. Here is why fish forgetting to be afraid is not a small story. Portavella and the researchers working alongside him propose that this medial pallium is not some isolated, fish-only curiosity. It is proposed as homologous to the pallial amygdala found in every land vertebrate, your own included. Homologous is doing real work in that sentence. It does not mean fish independently reinvented something that merely resembles an amygdala. It means the working hypothesis, built from tracing connections, transmitters, and cell types, is that fish and land vertebrates most likely inherited this same basic and kept it running on two separate branches ever since. In land vertebrates, the amygdala complex is generally understood as the hub where incoming sensory information gets evaluated for survival relevance and tagged with the right bodily response, often faster than the same information reaches full conscious awareness. That general job description, evaluate for danger, tag it, keep the tag ready for instant use, is exactly what researchers are testing for when they go looking for anything resembling it in a fish, a lamprey, or a human patient with a rare kind of brain damage. Finding the same job being done by an anatomically comparable structure in animal after animal is a much stronger claim than finding a passing resemblance. This is not a one-species coincidence. In 2014, a separate team, Marion von Trotha, Philippe Vernier, and Laura Bally-Cuif, published a study on zebrafish, a different species entirely, in the European Journal of Neuroscience. They identified the same basic division, a region called DM, proposed as homologous to the mammalian amygdala, sitting near a second region, DL, proposed as homologous to the hippocampus. DM was not just anatomically similar to an amygdala. It was functionally active in exactly the situations you would expect from something amygdala-like, engaged during an innate anxiety-like response in a light and dark avoidance test, and described by the researchers as hosting the equivalent of the amygdala nuclei that drive avoidance strategies and avoidance learning specifically. Two different fish, two different research teams, a decade apart, and the same basic architecture keeps showing up, doing the same basic job. Researchers who compare species this far apart are testing a specific kind of claim. If two animals separated by hundreds of millions of years of independent evolution, with no recent shared ancestor doing anything remotely solution to close to the same problem, that suggests the solution was something close to inevitable, not a one-time historical accident. A goldfish, a zebrafish, and a person did not each sit down separately and invent a fear circuit from nothing. On the leading account here, they are separate descendants carrying forward one very old answer to one very old problem. How do you make sure a body remembers, instantly and completely, what almost killed it? How far back does this general layout go? In 2017, the researchers Shreya Suryanarayana, Brita Robertson, Peter Wallen, and Sten Grillner examined the brain of the lamprey in a paper published in Current Biology. They described the lamprey as the phylogenetically oldest group of extant vertebrates alive today, a jawless fish whose lineage split off from the rest of the vertebrate tree before almost anything else still swimming today did. Their paper describes this general layered brain architecture, the blueprint that eventually becomes cortex in land vertebrates, as carrying what they call a long-standing evolutionary precedent on the order of some 500 million years. That paper is about the broader architecture of this kind of brain tissue, not a specific, but it sets the right scale. Whatever is running in a goldfish's medial pallium is not a recent addition bolted onto an otherwise modern brain. It sits on hardware whose general blueprint was already old before most of the animal kingdom you could picture had evolved into anything resembling its current shape. None of this on its own tells you anything about your own body. So look at what happened to a human patient in a completely unrelated experiment on a completely different species, run more than 2 decades. In 1995, the researchers Anton Bechara, Daniel Tranel, Hannah Damasio, Ralph Adolphs, Charles Rockland, and Antonio Damasio published a study in the journal Science. They worked with patients who had rare, selective, symmetrical brain damage and ran an experiment carrying almost the same logic as the goldfish test. A colored slide meant nothing on its own until it was reliably followed by a sudden loud noise, and the researchers measured a small, automatic electrical change in the skin that shows up when a person becomes afraid, whether or not they say anything about it. One patient had damage limited to the amygdala. That patient never developed the fear response to the slide. The skin stayed quiet, pairing after pairing. But the same patient could describe in plain language exactly which color had been paired with the noise. The facts got in. The fear never did. The second patient had damage limited to the hippocampus instead and showed close to the opposite pattern, a fear response building normally. A measurable reaction in the skin every time the color appeared with no memory at all of why. In the researcher's own framing, one patient acquired the facts but not the fear. The other acquired the fear but not the facts. Set the goldfish study and the human study side by side and the shape is close to identical. One specific piece of tissue, and only that piece, is what allows a warning signal to convert into a lasting, automatic bodily state of fear. A separate neighboring piece handles the plain facts of what happened, and either piece can be knocked out without touching the other. That structure was not built once by one species for one purpose. It shows up in a fish moving through a shallow tank and in a person lying under a hospital monitor, arrived at, on the leading evidence, through the same evolutionary process branching out from a single, much older point of origin. Monitor to notice a version of this in yourself. Think of a fear you carry that arrives whole and immediate and ask whether the specific memory that built it arrives just as fast. For a lot of people, the fear gets there first, complete and already running. The explanation, if it shows up at all, arrives late and it does not always show up. None of this proves what a goldfish or a zebrafish experiences when that circuit fires, and it is not offered as a claim about animal consciousness in general, which stays a genuinely open question well past the reach of anyone's study. What it does establish is narrower and arguably stranger. The specific piece of machinery required to turn a warning into fear that sticks rather than a fact that is simply known is not something your species built recently or alone. It was already doing that exact job in a comparable form in animals whose branch of the family tree split from yours long before there were mammals to speak of. Which means the version running in you right now, the one that makes a specific street feel wrong before you consciously remember why, or tights your chest at a sound you cannot immediately place, is not a loose metaphor borrowed from older animals for a nicer story, depending on exactly which piece of your temporal lobe is doing the work at that moment. Same specific system, running the same specific job it has been running for a very long time. Here is the question for the comments. Think of something that makes your body react before you can consciously place why. Do you have the plain fact behind it ready to hand, or only the feeling with the reason still missing? I read these. In the next episode, we look at animals that have never once encountered a human being in their entire lives and already know on first contact to run.